Catalase was first discovered in 1811 by Louis Jacques Thenard, when he determined that an unknown substance was responsible for the decomposition of hydrogen peroxide. He was also the one that discovered hydrogen peroxide. And catalase was named in 1900 by Oscar Lowe and was first crystallized in 1937 by James Sumner and Alexander Dones. Catalase catalyzes the decomposition of hydrogen peroxide to water and oxygen, and this is a very important reaction because it turns hydrogen peroxide, which is a harmful byproduct of metabolism that can cause tissue damage, into the non-toxic byproducts oxygen and water. So catalase functions to protect cells from oxidative damage. And catalase is found in basically anything that requires oxygen. It is found in plants and animals, and on a cellular level, catalase is found in high concentrations in cells' peroxisomes, and catalase is especially concentrated in organs with high metabolic activity, such as the liver. So a little bit about the structure of catalase. Catalase is a tetramer, so it has 4 subunits. Each of the 4 polypeptide chains is approximately 500 amino acid residues long, and catalase contains 4 porphyrin heme groups. The porphyrin heme groups form part of the active site that interacts with hydrogen peroxide. And here is a picture of catalase. Hopefully you can make out the 4 subunits. So the reaction that catalase catalyzes is 2 hydrogen peroxides into 2 water molecules and oxygen. So this mechanism of catalase is on your handouts. The structure on the bottom left is where this begins. So the mechanism of catalase begins with a general base catalysis. When a hydrogen peroxide enters the active site of catalase, histidine grabs the hydrogen from the hydrogen peroxide, resulting in a nucleophilic attack of the hydrogen peroxide onto the heme group. The water molecules hydrogen bonding to the glutamine 168 and aspartic acid 128 are no longer interacting with the hydrogen peroxide, which is what the next structure is showing. Aspartic acid 348 then grabs the hydrogen from histidine 128, and the resulting lone pair on the nitrogen moves to the adjacent carbon, and the double bond abstracts the hydrogen from arginine 354. And simultaneously, the heme group pushes electrons onto the oxygen of the hydrogen peroxide, breaking the oxygen-oxygen bond of hydrogen peroxide. And then the water dissociates, which is what the next one is showing, basically the same thing without the water. A new hydrogen peroxide molecule comes into the active site, and the original oxidation state of the heme group is regenerated by hydrogen shifts and the formation of O2. And the arrows about to be drawn are showing the movement of electrons. The oxygen on the heme group grabs the hydrogen from asparagine 148, followed by electron movement. And also the nitrogen of arginine 354 abstracts a hydrogen from histidine 128. And electron pushing results in histidine 128 abstracting a hydrogen from aspartic acid 348. The active group is regenerated and available to start the process over. So the activity of catalase can easily be assayed. Since catalase reacts specifically with hydrogen peroxide, all you need is some hydrogen peroxide and a source of catalase. We chose to use liver since catalase is especially concentrated in liver and will have a more visible effect. We just used 3% hydrogen peroxide from Harris Teeter and some beef liver as our source of catalase. And as you can see, the reaction was pretty quick. The bubbles are forming as oxygen is being produced from the decomposition of the hydrogen peroxide. So there you go, the action of the all-important enzyme catalase.